Hi, my name is Anuradha Surendra and I'm with the National Research Council Canada. My poster is titled Metabonit, an application and database for small molecule protein interactions. Our aims with Metabonit were threefold. 1. Provide a systematic understanding of small molecules and protein relationships. 2. Compile a database of available interactions from publicly available information. And 3. Generate an interactive website by linking the compiled information for query with seamless information generation by the end user. Accordingly, a workflow was produced and can be simplified into 3 parts: collation of public data, creation of relational databases, and the generation of a web-based application. Small molecule, or small molecule protein interaction data was parsed using R and Python scripts from 4 different publicly available databases. 13,566 drug interaction data from DrugBank, 114,187 metabolite interactions from HMDB, 21,382 protein interactions from PDB-Bind, and 4,341 protein interactions from Brenda were pulled and incorporated into Metabonit. Furthermore, corresponding information from PubChem, UniProt, and PubMed were extracted. The Postgres database was split into separate tables for each collated dataset. These tables incorporate details such as compound name, gene ID, gene name, UniProt ID, and PubChem ID. The PubChem, UniProt, and PubMed tables provide detailed information on various properties. These relational databases were connected by unique identifiers such as PubChem IDs, UniProt IDs, and PubMed publication IDs. The interactive website presenting the database to the end user was mainly built using RShiny and incorporating various R libraries. The user is able to visualize the compound, drug, or metabolite and gene relationship through network graphs. For each chemical or gene chosen through the drop-down menu, the R application provides comparative network plots. One application of Metabonit is in the analysis of protein interactions. Remdesivir is of great interest as a possible treatment for COVID-19. Through observations from DrugBank, Remdesivir has 2 protein targets: POC6X7, replicase polyprotein 1AB, and QO5318, RNA-directed RNA polymerase L. Both of the above targets affect RNA transcription. Ribavirin has 770 drug-drug interactions within DrugBank. The top 4 classifications for the drug-drug interactions are 26 proteins, 21 amino acids, peptides, and proteins, 10 biological factors, and 10 peptides. Metabolite protein interactions can be viewed as an alternative way for analyzing metabolomics or genomics results. Glutathione is a 3-peptide metabolite with a major role as an antioxidant and a regulator of numerous cellular processes in disease. Analysis of glutathione partner interactions obtained from HMDB data shows 57 protein associations. These associations can be classified by the following: 41 enzymes, 11 unknown, and 5 transporters. By combining published drug interaction data, the Metabonit database and the application interface provides an overview of drug, metabolite, and protein interactions. The application allows the user to graphically visualize relationships between small molecules and proteins. The future goal is to supplement the database using natural language processing for parsing more information from publications. To highlight the usage of Metabonit, I will walk you through 2 different examples. But before that, this is the graphical user interface for MetaboNet. Within the main panel, there are 3 different drop-downs. The first is to select the different databases. The second is to select the different type of interactions to display. And the third is to select the metabolites or proteins you would like to display. The network graph is displayed within the Plot tab, and a summary table with the selected metabolite or protein and their properties is displayed within the Summary tab. For my first example of metabolite-protein interactions, we'll take the example of glutathione. This is the network graph for glutathione. Within this drop-down, we can select whether we want to look at the enzymes that interact with glutathione, transporters, or the unknown proteins that interact with glutathione. For my second example, we'll look at the protein metabolite interactions of 3 proteins, NT5E, NT5C, and NT5M. Here we have the network graph for those 3 proteins. When you hover over the yellow nodes, you can see the metabolites that interact with these 3 proteins highlighted. I hope this gives you a better understanding of what we're trying to achieve with MetaboNet. Thank you for coming and listening to my poster presentation.